So today we'll be starting off with the effect of enzyme concentration on rate of enzyme activity. Yesterday we talked about two factors. The first one was temperature and the other one was the pH. Now figure 3.11a shows the result of an investigation using enzyme catalase again and its substrate hydrogen peroxide. What is the enzyme catalase that breaks down H2O2 into two products, O2 and H2? वही एग्जांपल बार बार यहाँ पे यूज हो रही है. The catalase is present in an extract made from celery, and different concentrations of catalase solution were added to hydrogen peroxide solutions. The different concentrations were prepared by varying the initial volume of celery extract and then making up to the standard volume with distilled water. The quantity of the hydrogen peroxide used was the same at the start of all five reactions. Distilled water क्यों डालते हैं? Distilled water is something that is used to make something dilute. What's the difference between the terms dilute and concentrated, according to your paper number 3?
Concentrated means that it has a higher amount of the substrate or the solution we are using, and diluted means that it is mixed with water
Exactly.
or with any other.
बिल्कुल solute कम है, water ज्यादा हो तो वो dilute होता है. You can see that the shape of all five curves is similar. ठीक है, यहाँ पे you can see over here. One of them were 4 centimeter, 3 centimeter, 2 centimeter, 1 centimeter, 0.5 centimeter cube. Now you can see that as the volume of the extract increases, what is happening? आपके पास the rate of reaction is also increasing, which tells us that greater the concentration of enzyme, the greater would be the faster would be the reaction, because more enzyme is available, more substrate would be react, and there won't be any waiting time. कल हमें क्या issue हो रहा था temperature pH में, that there was a waiting time. Right now there is no waiting time because you have a lot of enzyme. In each case, the reaction begins very quickly as a steep curve, and then it gradually slows down and the curve levels off. In order to look at the effect of enzyme concentration on reaction rate, you must compare the rates of these five reactions, and it is best to look at the rate right at the beginning of the reaction. This is because once the reaction is underway, the amount of substrate in each reaction begins to vary because substrate is converted to product at different rates in each of the five reactions. आपको gradient से पता लग रहा है कि सबका डिफरेंट रेट था. हाँ, सेम कहाँ पे था? अगर आप वही दोबारा से फर्स्ट 30 सेकेंड्स में देखो, दिस इज द बेस्ट 30 seconds to measure the rate of reaction because the rate is the fastest and the steepest in the first 30 seconds. Overall हमारे पास एक directly proportional relation निकला between the concentration and the rate of reaction. As the concentration increases, the rate of reaction also increases. By calculating the initial rates, you can be sure that the differences in reaction rates are caused only by the differences in enzyme concentration and not by the substrate concentration. To work out the initial rate for each enzyme concentration, you can calculate the slope of the curve 30 seconds after the beginning of the reaction, as explained in P1. Ideally, you should be using this at an earlier stage of the reaction. बिल्कुल जैसे reaction start होता है उस वक्त the rate is the highest, the rate is the steepest because you're getting all the optimum conditions. उसके बाद आहिस्ता आहिस्ता what happens that if you delay it more than 30 seconds, substrate is no more available. बेशक enzyme होंगे, substrate won't be available. So you can then plot a second graph showing the initial rate of reaction against enzyme concentration. The graph shows that the initial rate of reaction increases linearly. Linearly मतलब directly proportional relation होता है to the enzyme concentration. Because if one directly proportional means when one variable increases, the other variable also increases. Or if one variable decreases, the other one also decreases. This is just what common sense says should happen. The more enzyme is present, the more active sites will be available for the substrate to slot into. As long as there is plenty of substrate available, the initial rate of reaction increases linearly with the enzyme concentration. So the effect of substrate concentration on the rate of enzyme activity. हमने enzyme concentration तो बढ़ा दिया. Now we are going to increase or decrease the substrate concentration that has to be actually broken down. So this is another figure abhi aage aayegi that shows you the increase in substrate concentration and the initial rate of reaction. ये directly proportional नहीं है. There's a point यहाँ पे curve भी हो रहा है. Why didn't enzyme concentration curve? क्योंकि over there enzymes don't have to wait. Over here the substrate is going to have to wait because now enzyme would be in a limiting amount and substrate would be in excess. क्या फायदा होगा आपको सबस्ट्रेट एंजाइम बढ़ाने का फायदा है? सबस्ट्रेट बढ़ाने का फायदा होगा लेकिन एक हद तक होगा, क्योंकि अब एंजाइम भी तो साथ बढ़ाने पड़ेंगे ना। दैट इज वाई एंजाइम कॉन्सेंट्रेशन इज टोटली लीनियर स्ट्रेट लाइन। बट सबस्ट्रेट कॉन्सेंट्रेशन आप बढ़ाओ, ठीक है, अच्छी बात है, बट टिल व्हाट? देयर वुड बी एन एक्सटेंट टू विच यू कैन एक्चुअली इंक्रीज द सबस्ट्रेट कॉन्सेंट्रेशन। एक पॉइंट आएगा कि सबस्ट्रेट डाले जा रहे हो, एंजाइम है नहीं जो तोड़ेगा। As in the previous, now you are keeping the volume of catalyst constant. Now the enzyme is constant and you are increasing the substrate. So this is the first, this is what you can observe. And what are you going to do if you want to measure the rate of reaction? Again, you going to use the start of the curve. This is the gradient that you are always going to use to calculate the rate of reaction. We call these the initial rates of reaction plotted against substrate concentration. So as the substrate concentration increases, the initial rate of the reaction also increases, and this is what you would expect. The more the substrate molecules there are, the more often one will enter an active site. However, अब scene मुश्किल है. However, if you go on increasing substrate concentration, keeping the enzyme concentration constant, there comes a point when every enzyme active site is full. And if more substrate is added, enzymes cannot work faster. Enzyme अपनी speed से काम करेगा, आप पीछे से डालते रहो substrate. Enzyme will still work at their own speed. So substrate molecules are effectively queuing up for an active site to become vacant, and the enzyme is working at its maximum possible. We call this point as Vmax. Vmax क्या है? The point where enzyme is working at its maximum possible. We call it the Vmax. So can you define me what is Vmax?
Vmax is the point where the enzyme can work at its best.
The theoretical maximum rate of an enzyme control reaction obtained cup when all active sites of enzymes are occupied. So now we are going to compare the different enzyme affinities. Affinity means ability, the measure of the strength of attraction between two things. A high affinity means that there is a strong attraction, and when applied to enzymes, affinity is a measure of the strength of attraction between the enzyme and its substrate. The greater the affinity of an enzyme for its substrate, the faster it would work. जितनी ज़्यादा एक enzyme की affinity होगी, the more the affinity of an enzyme. What is affinity? Strength of attraction to the substrate. The more the enzyme has affinity for its substrate, which means the greater the attraction between enzyme and substrate, and greater would be the chance of enzyme substrate complexes being formed, which means they would work faster. Another way of thinking about this is to say that higher the affinity, the more likely it is that the product will be formed when a substrate molecule enters the active site. And if the affinity is low, the substrate may leave the active site before a reaction takes place. There is enormous variation in the speed at which different enzymes work. A typical enzyme molecule can convert about 1,000 substrate molecules into product per second. जैसे कि कार्बोनिक एनहाइड्रेज है, ये फास्टेस्ट एंजाइम है एवर नोन. Carbonic anhydrase can remove 6 lakh molecules of carbon dioxide from respiring tissue per second, which is like 10 to power 7 times faster as कि वो अगर एंजाइम के बगैर वो हो रिएक्शन तो वो एक मॉलिक्यूल जाए. ये 10 लाइक एक कितने जीरो है ये 10 to power 7 times more faster reaction है as it would have been done without an enzyme. So it has a very high efficiency. तो बिल्डअप ऑफ कार्बन डाइऑक्साइड लीथल भी बहुत होता है टिश्यूज के लिए. आपकी बॉडी में कार्बन डाइऑक्साइड बिल्ड हो जाना, आपके ब्लड में डीऑक्सीनेटेड ब्लड बहुत ज्यादा आ जाना कैन लीड टू फटीग एंड मेनी अदर प्रॉब्लम्स. दैट इज व्हाई ये एंजाइम बहुत एफिशिएंट है और कुदरत का निजाम है कि ये एंजाइम अपने काम में बेस्ट है. सबसे अच्छा काम ये करता है दैट इट रिमूव्स कार्बन डाइऑक्साइड एज सून एज पॉसिबल फ्रॉम द बॉडी. In the previous section, you saw that a useful indicator of efficiency of an enzyme is Vmax. This tells you the maximum speed at which an enzyme works. And remember that V stands for velocity, which means speed. तो maximum speed समझ लो जो हमने इधर पीछे पढ़ा था. At Vmax, enzyme molecules are bound to substrate. All active sites are busy. The enzyme is saturated with substrate. Saturated मतलब busy. All the active sites are full, busy. And Vmax can be measured in the way described in figure 3.1. The initial rate of reaction is measured at different substrate concentration while keeping the enzyme concentration constant और वही सारा कुछ आपके पास आ गया. The initial rate for each substrate concentration is plotted against substrate concentration, producing a curve like those shown. Unfortunately, एक point आता है that the curve flattens out as well, but बिल्कुल exact straight line नहीं होगा. It's gonna keep on having a slope. You cannot ऑलवेज रीड द वैल्यू ऑफ वी मैक्स, क्योंकि कभी भी फ्लैट नहीं हो रहा। या तो ऐसा ना कि पॉइंट कहीं पे ग्राफ फ्लैट हो जाए तो मैं कहूँ यार ये, हाँ ये वाला पॉइंट जो है ना ये वी मैक्स है। सो देर इज अ मैथमेटिकल वे आउट ऑफ दिस प्रॉब्लम। फ्रॉम द डाटा इन सच ग्राफ इट इज पॉसिबल टू कैलकुलेट हाफ वी मैक्स। आप हाफ वी मैक्स निकालो। फुल वी मैक्स कैलकुलेट करना पॉसिबल इसलिए नहीं क्योंकि लाइन कभी फ्लैट नहीं होगी, ये चलती रहेगी। फ्लैट तो तब होगी जब लाइक सबसेट मुक जाए, या सबस्ट्रेट तो मुक ही नहीं रहा यहाँ पे, ठीक है। फ्लैट लाइन तो तब होगी व्हेन सबस्ट्रेट फिनिशेस। यहाँ पे हम सबस्ट्रेट ही तो बदल रहे हैं, एंजाइम कांस्टेंट है। फ्लैट हाँ उस केस में हो सकती है अगर मैं एंजाइम डालना बंद कर दूं, वहां पे फ्लैट हो सकती है। लेकिन सबस्ट्रेट जब आप डाल रहे हो, रिएक्शन वुड नेवर इन लाइफ गो फ्लैट। वो इन्फिनिटी स्लोप, पूरी लाइफ वो स्लोप पे जाता रहेगा, क्योंकि एंजाइम इज नेवर इट सेल्फ यूज्ड अप। समझ आई? गॉट इट?
या
दैट इज वाई वी कैन नॉट कैलकुलेट द वैल्यू ऑफ वी मैक्स। लेकिन हाफ वी मैक्स निकाल सकते हो। हाफ वी मैक्स इज एग्जैक्टली हाफ द मैक्सिमम वेलोसिटी। इट इज जस्ट एज यूजफुल एज वी मैक्स एज एन इंडिकेटर ऑफ हाउ फास्ट एन एंजाइम वर्क्स। तो हाफ वी मैक्स आप एक तुक्के से कहते हो कि यार दिस वी मैक्स वुड बी समवेयर अराउंड हेयर, या आपको क्वेश्चन में दी होगी। हाफ वी मैक्स वैल्यू वुड बी एग्जैक्टली द हाफ ऑफ दैट, ठीक है, जहां से जितना वो डिस्टेंस था। और वहां से यू आर गोइंग टू चेक द सबस्ट्रेट कॉन्सेंट्रेशन, कि कितनी सबस्ट्रेट कॉन्सेंट्रेशन के ऊपर वी मैक्स था। दैट वुड बी गिविंग यू एन आंसर फॉर योर के एम। वो आपको देगा आपका के एम। के एम इज दैट सबस्ट्रेट कॉन्सेंट्रेशन, माइकलिस मेंटन कांस्टेंट ऑफ एन एंजाइम, वो सबस्ट्रेट कॉन्सेंट्रेशन एट विच द एंजाइम वर्क्स हाफ इट्स मैक्सिमम रेट। हाफ वी मैक्स के ऊपर जो कॉन्सेंट्रेशन वैल्यू थी उसको हम कहते हैं के एम, which is the substrate concentration at which an enzyme works at half of its maximum rate, used as a measure for the efficiency of an enzyme. The lower the value of Km, the more efficient is the enzyme. And if you think about it, the higher the affinity of an enzyme for its substrate, the lower the substrate concentration needed to reach as well. जितनी ज्यादा अगर आपको higher the affinity चाहिए, lower would be the substrate concentration, थोड़ी ही substrate concentration में आपकी आपका Vmax रीच हो जाएगा। हाई एफिनिटी, कम सबस्ट्रेट कॉन्सेंट्रेशन चाहिए होगी, Vmax जल्दी आ जाएगा, मतलब हाफ Vmax जल्दी रीच हो जाएगा। इज इट क्लियर?
यस।
नाउ वी विल मूव ऑन टू एंजाइम इनहिबिटर्स। ठीक है, कैन यू टेल मी व्हाट इज द मीनिंग ऑफ इनहिबिट?
नो, नॉट दैट क्लियर।
कब्जा कर लेना, ठीक है, कब्जा करना। और टेकिंग ओवर, कंपटीशन देना, ठीक है। इनहिबिट करना, किसी और की जगह पे आ जाना। गॉट इट? इन्वेड।
यस।
अब एंजाइम के इनहिबिटर्स भी होते हैं, ठीक है। कंपेटिटिव रिवर्सिबल इनहिबिशन का क्या मतलब है? एक और मॉलिक्यूल आया है, कंपटीशन दे रहा है, लेकिन आप उसको हरा सकते हो, इट इज रिवर्सिबल। As you have seen that the active site of an enzyme fits its substrate perfectly, and it is possible, however, for a molecule with it similar in shape to the substrate to enter an enzyme's active site. You have an enzyme which has an active site like this, ठीक है, and now there's a substrate molecule that has to come on this active site, लेकिन there is a exact similar molecule that has the same exact shape like this substrate। ये substrate है, लेकिन exact shape का एक inhibitor आ जाएगा जो substrate की जगह आके यहाँ पे लग जाएगा। competition दे रहा है। If an inhibitor molecules bind only briefly to the site and comes out again, वो competition है, लेकिन it is reversible। अब कंपटीशन दो किस्म से मिल सकता है। ये इनहिबिटर आया रिवर्सिबल की जगह, ठीक है। दिस इनहिबिटर केम इन प्लेस ऑफ द सबस्ट्रेट फॉर ओनली अ शॉर्ट पीरियड ऑफ टाइम, only for a short period of time, gave competition, but for a short period of time, which means it was reversible. But if it is permanent, it is a competitive inhibition, but it would be irreversible. Got it?
या
अब how can you make it reversible? If there is more of the substrate present than an inhibitor, अगर आपका substrate ज्यादा है inhibitor से and inhibitor कम है तो reversible हो जाएगा। inhibitor थोड़ी देर के लिए ही आएगा। कंपटीशन दोनों दे रहे हैं, फर्क ये है कि एक को सबस्ट्रेट हरा गया है, तो वो इनहिबिटर थोड़ी देर के लिए लगा रहा। इधर सबस्ट्रेट की कॉन्सेंट्रेशन कम थी, इनहिबिटर को नहीं हरा सका और इनहिबिटर परमानेंट आ गया। कंपटीशन दोनों ने दिया, बट वन वाज रिवर्सिबल, वन वाज इररिवर्सिबल। Now the molecules can easily win the competition if the substrate is higher and inhibitor is lower. and so the enzyme's function is more or less unaffected. Unaffected हुआ है, कम unaffected हुआ है. However, if the concentration of the inhibitor rises, तब भी competition देगा, but it would be irreversible. It would become less and less likely that the substrate will collide with an empty active site और उसको मौका ही नहीं मिलेगा, तो वो irreversible के अंदर तब्दील हो जाएगा. We call it competitive inhibition. It is said to be reversible, not permanent, because it can be reversed by increasing the concentration. आप फिर भी इसको रिवर्स कर सकते हो, कैसे? Substrate की concentration बढ़ा दो. Increase the concentration of the substrate so it can fight the competitive inhibitor. फिर एक टाइप आती नॉन competitive inhibition as well, जो competition दे ही नहीं रही है. फॉर एग्जाम्पल, पहले एक कंपेटिटिव इनहिबिशन की टाइप पढ़ते हैं. इट अकर्स इन द ट्रीटमेंट ऑफ अ पर्सन हु हैज ड्रंक एथिलीन ग्लाइकोल. एक बंदा है जिसने एथिलीन ग्लाइकोल पी ली है, और ये एग्जाम्पल पेपर में आ चुकी है, मुझे याद आ गया. Ethylene glycol is used as an antifreeze and is sometimes drunk accidentally. Ethylene glycol is rapidly converted the body into oxalic acid, which can cause irreversible kidney damage. However, the active site of the enzyme which converts ethylene glycol to oxalic acid will also be kept ethanol if the poisoned person is given a large dose of ethanol. अब बंदे को अगर आपने रिवर्स करना है उसका इफेक्ट, तो आप क्या करोगे? आप उसको लार्ज डोज ऑफ एथेनॉल दे दो. अगर आपने उसका डोज का इफेक्ट कम करना है, क्योंकि एथेनॉल अंदर जाएगा, बॉडी में एथिलीन ग्लाइकोल की कंपटीशन देगा, और वो ऑक्सैलिक एसिड में कन्वर्ट करने ही नहीं देगा. गॉट इट?
Yeah.
थोड़ी देर बाद बॉडी में एथिलीन ग्लाइकोल किसी काम का नहीं रहेगा, तो वो बॉडी से फिर एक्सक्रीट हो जाता है. देन व्हाट इज दिस नॉन कंपेटिटिव रिवर्सिबल इनहिबिशन? नॉन कंपेटिटिव, कंपटीशन दे ही नहीं रहा. ठीक है, ये एक्टिव साइट है, ये सबस्ट्रेट मॉलिक्यूल है, ये आया आपके पास नॉन कंपेटिटिव इनहिबिटर. ये सबस्ट्रेट को कहे तू जा लग जा, एक्टिव साइट है, मैं कंपटीशन दे ही नहीं रहा तुझे. क्यों? मैं जाऊंगा एक और तरफ से, मैं एक और जगह से जाके अटैक करूंगा. अ डिफरेंट काइंड ऑफ रिवर्सिबल इनहिबिशन इज पॉसिबल विच इज नॉन competitive. Non competitive ones do not bind to the active site. They bind to another site which is known as allosteric site. Allosteric site वो होती है all other sites other than the active site. In this type of inhibition, the inhibitor molecule binds to another part of the enzyme, not the active site. While the inhibitor is bound to the enzyme, it can affect the normal arrangement. लेकिन ये करेगा क्या? This is the enzyme. नॉन competitive इनहिबिटर जाएगा, एक्टिव साइट पे बाइंड नहीं करेगा. कहाँ जाएगा? कहीं और बाइंड कर जाएगा, यहाँ पर. अब आप कहोगे कोई बात नहीं, कर ले, सबसेट आके तो अपना काम कर लेगा. नहीं. The moment this inhibitor binds on the other side, it disorients this bond arrangement as well. खुद भी आके लग गया, और जो आ रहा था अच्छा खासा, जिसको आप कंपटीशन नहीं भी दे रहे थे, उसका भी आपने काम खराब कर दिया है. गॉट इट?
Yeah.
सो दोनों तरह से, कंपेटिटिव हो तो भी सबस्ट्रेट के लिए मुश्किल है, और नॉन कंपेटिटिव हो, बेशक सबस्ट्रेट के लिए जगह खाली है, लेकिन काम तो खराब वो पीछे से आके कर दिया उसने. बिकॉज देर आर मेनी हाइड्रोजन बॉन्ड्स एंड हाइड्रोफोबिक इंटरेक्शंस दैट होल्ड द एंजाइम मॉलिक्यूल in its 3D shape, and that results in distortion, changes the shape of active site, and it also therefore inhibits the ability of the substrate to enter the active site. While the inhibitor is attached to the enzyme function is blocked, and the substrate molecule and the inhibitor are not competing for the active site. We call this non-competitive inhibition. आपस में compete नहीं कर रहे, लेकिन वो उसका काम खराब कर गया आके. अब आप बेशक सबस्ट्रेट की concentration बढ़ा लो, can this be reversed? No, because enzyme has changed its active site shape. Inhibition of an enzyme can be harmful or even fatal as well. But sometimes inhibition essential भी है. जैसे आपने वो डोज कम करनी थी, डोज कम करने के लिए इनहिबिशन ने आपको फायदा भी पहुंचाया. आप एक दवाई की अगर डोज कम करना चाहते हो अपनी बॉडी में, प्रोवाइड इनहिबिशन, ताकि रिएक्शन ना हो. लेकिन कभी कभार इट कैन बी हार्मफुल एज वेल. फॉर एग्जाम्पल, अगर कार्बोनिक एनहाइड्रेस वाली जो हमने एग्जाम्पल पढ़ी थी, अगर वो ना होता प्रेजेंट, तो आपकी बॉडी में CO2 ना जमा हो जाता, आप मर जाओगे. फॉर एग्जाम्पल, मेटाबॉलिक रिएक्शंस मस्ट बी कंट्रोल्ड, and no enzyme can be allowed to work without stopping at some point. Otherwise, more and more product would be constantly being made. So sometimes inhibition is good as well, sometimes it is bad as well. So can you give me a brief difference between both of the types of inhibition that we have studied? Tell me.
In competitive, the inhibitor um, takes place of this on this active side of the substrate, so the substrate cannot bind to it. While in non-competitive, the inhibitors take place on another side of the enzyme, but it changes the bonds of hydrogen, I guess, so that the active site changes in shape.
Good. When a substance reduces the rate of activity of an enzyme by competing with the substrate molecules for the enzyme's active site, increasing the substrate concentration reduces the degree of inhibition, and increasing the inhibitor concentration increases the degree of inhibition. Non-competitive inhibition is when a substance reduces the rate of activity of an enzyme, but increasing concentration of substrate won't help you anymore. वहां पे तो आपने substrate की concentration बढ़ा के काम सही कर लिया था, reverse कर लिया था. यहाँ पे you can't do it. One way of controlling metabolic reaction is also to use the end product of a chain. एक हमारे पास टाइप होती है end product inhibition. What is end product inhibition? ये substrate था, वो product 1 बना, product 2 बना, जो product 3 बना ना, वही वापस गया और सबस्ट्रेट के पास जाके इनहिबिट कर दिया कि बस मैं बन गया हूं यहाँ पे, मजीद प्रोडक्ट 1 ना बनाओ वरना मैं दोबारा और बनता जाऊंगा यहाँ पे. दिस इज नोन एज एंड प्रोडक्ट इनहिबिशन. आखिर वाला प्रोडक्ट वापस आके खुद करे कि मजीद प्रोडक्ट 1 ना बनाओ. वी कॉल इट एंड प्रोडक्ट इनहिबिशन. नेक्स्ट वी हैव immobilizing enzymes. ठीक है, before that let me show you something regarding inhibition and then we'll start with our last topic which is the immobilization of enzyme. अभी तक क्लियर हो गया? एवरीथिंग ऑल क्लियर?
Yeah.
ओके.
Have you ever noticed how, when learning more about a science topic, we can get so interested that we start to discover favorites? Favorite dinosaur? I've found quite a few opinions there. Favorite animal? I'm a little obsessed. Favorite flower? Well, sometimes a little too obvious. Of course, you can always get a little more unique. Favorite protist? Favorite amino acid? Your favorite nitrogenous base? I'm a little saddened that no one has yet asked me those 3 above, nor have they asked me what I've been waiting to be asked, and yet have often volunteered. What is your favorite enzyme? ATP synthase is mine, but it's a hard choice because there are a ton of fascinating enzymes. We already have an intro video about enzymes, but this video topic is about to go into a little more. Enzyme examples, cofactors and coenzymes, inhibitors, and feedback inhibition. Enzyme examples are important because without learning about some real-life examples, it's kind of hard to connect to how important they are to living things. Let's start with some fascinating enzyme examples in the human body. A great place to focus on digestion. Many enzymes focus on breaking down certain biomolecules. You remember the 4 biomolecules: carbohydrates, lipids, proteins, nucleic acids. These biomolecules are found in your food. You've got to break those biomolecules down in digestion. Amylase is an example of an enzyme that breaks down carbohydrates, and one action location is in the mouth. Amylase can break the glycosidic linkages in starch, which is a large carbohydrate, into smaller carbohydrates. Lipase is an example of an enzyme that breaks down lipids, and one action location is in the small intestine. Lipase can break the ester bonds found in triglycerides, which are a type of lipid, into their building blocks: fatty acids, and glycerol. Pepsin and trypsin are some examples of enzymes that break down proteins. An action location for pepsin is in the stomach, and an action location of trypsin is in the small intestine. These enzymes break the peptide bonds in proteins. Pepsin breaks down proteins into peptides. You even have nucleases that break down nucleic acids. That's DNA and RNA. And yes, DNA and RNA can be found in your food. Nucleases break down phosphodiester bonds found in connected DNA and RNA and helps break them into nucleotides. By the way, once all of these biomolecules are broken down into smaller pieces, there are even more digestive enzymes that can break them down even more. But it's not just digestion. You can name any human body system: excretory system, respiratory system, circulatory system, and you'll find there are enzymes involved. These enzymes might break down or build up the substances that they act upon, which is needed by different body systems. And while you're at it, realize that enzymes are not just for humans. Enzymes are found in all living organisms, like this Venus flytrap here. Beyond the 3 domains of living organisms, you'll even find some types of enzymes in viruses. Okay, so moving on. What's next on our list? Oh yes, cofactors and coenzymes. So just a quick review from our main enzyme video. This is an enzyme here, and we have our active site and a substrate that could fit in the active site for the enzyme to act upon. It's an enzyme substrate complex when the substrate is bonded in this active site. And there is likely induced fit, where the active site actually changes its shape slightly for an ideal fit, an enzyme substrate hug. But the thing is, enzymes often don't do it alone, and they tend to have cofactors, which tend to be inorganic, like zinc or iron, or coenzymes, which tend to be organic, like many vitamins. Cofactors and coenzymes may be permanent or temporary, which often depends on how they're bonded. A cofactor or coenzyme may sit in this active site here. It helps the bonding of the enzyme and substrate so that it can work at its best, and sometimes it won't even work at all without them. An example of a cofactor or coenzyme? You know when we've mentioned DNA polymerase, an enzyme in DNA replication? It often has a zinc ion as a cofactor to help. There are also inhibitors. They might be reversible or not reversible. A lot of that depends on how they bond. They may be competitive or non-competitive. Competitive inhibitors tend to sit right here in the active site, blocking a substrate from being able to bind. The substrates are competing with the competitive inhibitor for binding. Non-competitive inhibitors bind to some other area of the enzyme, somewhere that isn't the active site. This can be referred to as an allosteric site, a site on the enzyme that isn't the active site. When the non-competitive inhibitor binds on the allosteric site, the enzyme has a shape change. If the substrate still binds, the enzyme's active site might not function well, or at all. You know, inhibitors might sound like a bad thing, but inhibitors can be harmful or they can be helpful. Some examples. You might remember DDT from our biomagnification video. It can act as an inhibitor for certain enzymes in the human body and can cause health issues. But understand that our body also uses inhibitors all the time to control a lot of biological processes. It's called feedback inhibition. This is a hypothetical example here, but let's say that enzyme 1 converts substrate A to intermediate B. Then enzyme 2 converts B to C. Then enzyme 3 converts C to a final product D. These enzymes are all working in a pathway here. But what if you have enough product D now? It's the ultimate product, but you have enough, and it's wasteful to keep producing it, or maybe it's even harmful to make too much. Well, product D might also be a non-competitive inhibitor for enzyme 1, stopping enzyme 1 from working, which will stop the process. Again, this could be reversible, or it could be that more enzyme 1s are produced in the body if this needs to start up again. So to recap, we explored some real-life enzyme examples, talked about cofactors and coenzymes, inhibitors, and feedback inhibition. But one last thing. We're big on asking why. What is the significance of learning this? So we know that enzymes are ubiquitous, love that word, and they are critical for understanding biological processes. They're also important to understand because many of the medications we use to treat diseases and disorders act on enzymes. One example, high blood pressure. It can be harmful to your organs and blood vessels. One class of blood pressure medications is the class of ACE inhibitors. ACE inhibitors work by blocking angiotensin-converting enzymes. When inhibited, these enzymes can't convert angiotensin to angiotensin II. Angiotensin II can be involved with raising blood pressure, so by blocking the enzyme that helps produce it, it can lower blood pressure. There's some more complexity with this to explore. By the way, it's not just the human body. Medications can act on enzymes and microbes that make us sick. For example, penicillin. Penicillin is an antibiotic, but it works by inhibiting the enzyme transpeptidase, which prevents many types of bacteria from being able to build their cell wall. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. Is it clear?
Yeah, it is clear.
Now let's talk about immobilizing enzymes. Enzymes have an enormous range of commercial applications, for example, in medicine, food technology, and industrial processes. Enzymes are expensive, and no company wants to have keep buying them over and over again. तो वो चाहते हैं जिस तरह से मर्जी रिसाइकल होते रहे. One of the best ways of keeping the cost down is to use immobilized enzymes that are fixed in some way to prevent them from diffusing freely in a solution. Immobilized enzyme कौन से होते हैं which have been fixed to a surface or trapped inside beads of agar gel. The enzyme lactase can be immobilized using alginate bead. आप उस एंजाइम को ना बीड्स के अंदर या तो सील कर दो. प्रिजर्वेशन नहीं होती, जैसा आप समझो प्रिजर्व कर रहे हैं हम एंजाइम्स. We are kind of preserving the enzymes. जैसे कि the substrate of lactase is in the disaccharide sugar lactose. Milk is allowed to run through the column of lactase containing beads and the lactase hydrolyze the lactose in the milk. अब आपने ना ऑलरेडी कुछ लैक्टोज के बीड्स इमोबिलाइज एंजाइम्स बनाए हुए हैं. आप क्या करते हो कि मिल्क को यहाँ से पास करते हो. मिल्क के अंदर क्या होता है लैक्टोज, और इन बीड्स के अंदर प्रिजर्व्ड लैक्टेज पड़ा हुआ है. तो वो इजीली ब्रेक डाउन इसको कर लेता है, और जो यहाँ पे प्रोडक्ट निकलता है दैट इज द ऑलरेडी ब्रोकन डाउन लैक्टोज, क्योंकि वो पास होता हुआ किस चीज में से आ रहा है, लैक्टेज में से. That is how your lactose-free dairy products are also made for those people who cannot digest lactose. You can also see that enzyme immobilization has several obvious advantages compared with just mixing up the enzyme with its substrate. If you just mix lactase with milk, you would have a very difficult job to get the lactase back again. And not only would you lose the lactase, but you would also have milk contaminated with the enzyme. Using immobilized enzymes mean that you can keep and reusing the enzymes and that product is enzyme free. Another advantage of this process is that immobilized enzymes are more tolerant to temperature. तो और अच्छी बात होगी ना कि वो सिर्फ रीयूज होंगे, बल्कि वो टेम्परेचर की टॉलरेंस उनके अंदर और पीएच चेंज की टॉलरेंस भी ज्यादा हो गई है. वरना जो नॉर्मल एंजाइम्स होते हैं दे आर वेरी सेंसिटिव. अब इसकी क्या वजह है? दिस मे बी पार्टली बिकॉज देयर मॉलिक्यूल्स आर हेड फर्मली इन शेप बाय एल्जिनेट. उनको पकड़ के रखा हुआ है ना एक चीज ने, वो बीड्स के अंदर बंद है. किस चीज की बीड्स में बंद है? एल्जिनेट की कवरिंग है उन बीड्स की जिसने उसको पकड़ के रखा हुआ है. सो इट मे ऑल्सो बी बिकॉज द पार्ट्स ऑफ द मॉलिक्यूल्स दैट आर एम्बेडेड in the beads are not fully exposed to the temperature or the pH changes. This is the one of the practical that you can just read. ठीक है. And that is the whole summary जो आपने read करनी है. And this is the end of your chapter. Now you are to submit me all of these exam style questions by tomorrow. Question number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 questions हैं. ठीक है. There are total of 12 end of chapter questions. Do you have this book?
Yeah, I have.
You have to solve all these 12 questions before 9 p.m. tomorrow. Got it?
Okay. Yeah.
Our class will start at exact 9:40. आज थोड़ा सा लेट हो गया तो नेट के बहुत इश्यूज है पाकिस्तानी क्या करें. तो
Yeah.
9:40 our class will start exact tomorrow. ठीक है, दोबारा लेट नहीं होगी. So I'll see you tomorrow then. I'll show you some lectures of biological molecules. Chapter 1 हो गया पूरा समझ आ गया. Cell structure, any problems?
No.